Here we are at viral taxonomy, and this part of viruses I no longer teach in the same level of detail I did when I made this video. So if you don't need to know viral taxonomy, skip ahead about 4 minutes in to viral phylogeny. Viral phylogeny is very important, and we will not skip that. So there is a lot of interesting viral taxonomy. Check it out if you want to, or if I've told you you have to, and learn it. If not, skip ahead to phylogeny. And the slide numbers will probably not match very well. We move on from virions to things like, how do we classify viruses? How are viruses related to each other? Viral taxonomy is based, as much as we can base it, on viral phylogeny. Remember now, taxonomy is classification. Phylogeny is evolutionary relationships. So phylogeny is how closely are we related to each other? When did our common ancestor exist? Classification is, what do we call it? So, or taxonomy is, what do we call it? So they group viruses into classes and orders and kingdoms and phylums and families and all that. It's the same groups as you see elsewhere, but they have different naming conventions. So all of those levels are italicized for some reason with the viruses, all the way up from like the family names. Those are the biggest names we are ever going to see, down to the genera, and we rarely use these terms, but I looked up SARS-CoV-2. What genus is it in? Well, it's a beta coronavirus, and all genera end in virus. All family names end in viridae. Family names, you'll see, you can learn something about the virus from them, like Hepadna viridae. These are all liver-infecting DNA genome viruses. Coronaviruses look like crowns under the electron microscope. And then within that genus, there would be species, and these would not be italicized. And the species for SARS-CoV-2 is severe acute respiratory syndrome related virus. So it's a descriptive name that tells you the description of the virus. Another species name, I think, is bacillus infecting virus, for a bacteriophage that infects bacillus. Yeah. And then strains within a species are the actual viruses. Like SARS is a strain within this species. SARS is a strain or a virus within that species. SARS-CoV-2 is a strain or virus within this species. And viruses don't mate with each other. There's no clear way to decide what should a species be, what should a genus be. I mean, you've probably been reading about mutations in SARS-CoV-2. There are lots of different versions of it in the world. If you are infected, there are lots of different versions of it inside you. As its nucleic acids are copied, it mutates. There are mistakes made. Should those all be species? I don't know. But they're not. That's not what virologists have decided to do. So how are viruses related to each other? Well, the first thing is they're not all related to each other. They don't all have a single common ancestor. What we can do by comparing viruses is see which ones are related to each other, which ones are descended from a common ancestor. And there are very big groups like that. But none of them have a place in here. No virus is related to a cellular life form. We don't know where viruses came from, but it is conceivable, kind of easy to imagine, that they are massive mistakes in gene replication or transcription. So they probably came from within a cell at some point, but they aren't descendants of a cell. They are a mistake that happened inside a cell. if anything. So this is an example of a viral phylogeny. These are RNA viruses. So all of these viruses have RNA genomes instead of DNA. We have DNA genomes, bacteria have DNA genomes. These viruses all have RNA genomes. This is what I was looking for, Orthomyxoviridae. I cut off the O. That's the influenza viruses and close relatives. Paramyxoviridae. Measles and mumps are over here. And this kind of tree, if you want to see how closely this is related to this, you measure all the distances in between them. And so we don't know where any of these came from, but they all have a common ancestor. So they probably are all descended from one virus. When we are describing viruses, there are some characteristics we have to think about. So one of them is the host range, and that is the spectrum of hosts the virus can infect. So like some viruses infect bacteria. They're called bacteriophage or just phage. Any cell on Earth has viruses that can infect it. That's a pretty safe thing to say. In fact, for every cell on Earth, there are roughly 10 virions. So in terms of like who outnumbers whom, viruses win that game. Yeah, there are an astronomically huge number of virions in the world. So some viruses infect a broad spectrum, like the rabies virus infects all mammals. Some influenza viruses can infect pigs and different bird species, humans, and others are much more specific. So some bacteriophage lambda only infects certain strains of Escherichia coli. I don't know what else to tell you about that. So I'm just saying some viruses are incredibly specific, like one strain within a species, and others are very broad. And that's their host range. Another thing we think about is tissue tropism, and this is the tissues that contain the infected cells. So what part of the body do they infect? What kind of cells do they infect? So mostly we think about this for animal viruses, and we can't easily figure this out usually, because when a person is infected with a virus, we can't like dissect them and find all the virus in their cells. But what we do know for things like SARS-CoV-2 is we know what receptor it uses to get into cells. And from there, we know what cells typically have that receptor on their surface, so then we can figure out where to expect that virus to go. In Unit 4, we will cover a lot of viruses as disease-causing agents when we think about the diseases, and COVID-19 is one we will cover. And what we will see is the different things that disease can do, the different symptoms we can have, can largely be predicted by where the ACE2 receptor is, like what cells have it. That helps us understand really why there can be different characteristics of the virus. So you'll see that in Unit 4. Today we're going to look at influenza viruses in some detail, and what they attach to is certain sialic acids, and there's not much more to say about that. So we've looked at host range, tissue tropism, and now we're going to look at genome types. And this is kind of a big one that we're going to come back to. So what we're talking about here is what kind of nucleic acid is inside the virion. What kind of nucleic acid is getting injected into a cell to reprogram the cell? And if it's DNA, that has consequences for what has to happen inside the cell to replicate the virus. If it's RNA, it's going to be a different process. So those are the two major options, but we can also see single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA. All of those exist. We have retroviruses, where you wouldn't know by looking at the virion that they are weird, but when you look at how they replicate, they are crazy, and they have RNA. So again, a virion is only going to have one of these. And when we think about single-stranded RNA viruses, in particular, these are very common. There are two big choices, or two big options for those. And for this, we should go back a second and think about basic cell biology. We have processes we need to do in our cells. We have DNA. Sometimes we have to replicate the DNA. We have to transcribe genes and make messenger RNA. And then that messenger RNA gets sort of read by ribosomes, which then make proteins. Well, messenger RNA is a single-stranded RNA molecule. And there are single-stranded RNA viruses that are basically messenger RNAs. So we call them positive sense or positive RNA viruses. And they act like messenger RNA. As soon as they get into the cytoplasm, they can very quickly get the cell to start translating their proteins, start making their proteins. For reasons I don't understand, a lot of viruses are negative sense single-stranded RNA. That means they don't directly encode proteins. We have to take the reverse complement of them to get a messenger RNA. In other words, a single-stranded RNA molecule of minus sense RNA would base pair to another piece of RNA, and that would be a messenger RNA. So if you have a plus sense single-stranded RNA and you look at the base pairing strand that attaches to it, that would be negative sense. And so there are viruses that have one or the other. There are some viruses that are positive sense and some that are negative sense. And we're going to look at a negative sense example in some detail today, just to show you, to give you a clearer picture of what is happening in cells. What do viruses do to the cells they infect? All kinds of stuff. So what I want you to do is choose from the next 3 slides, choose 5 of those effects that you understand. If you can look at antigenic changes on the cell surface and explain what that means in English, choose that one. Choose 5 of them that you could explain or define, and that's all I want you to know. I just want you to be able to tell people 5 different things a virus can do to a host cell. You don't need to explain how a virus does it, just explain what does it mean to release interferon. What does altered enzyme activity, what could that mean? What could host cell membrane permeability mean? So if you can find 5 of those that you understand, memorize them. And with that, I think I actually want to end this video, and the next one will be about viral replication, and that's kind of a bigger topic we have to think about. And that's when we really start to understand how viruses work. So I'll see you at that video, and we will talk about viral replication.